Hi, everyone. We are going to be doing a quiz for review. In the review, we will do some mutation questions. We will go through pedigrees as well as some monohybrid, dihybrid, and questions on codominance as well as incomplete dominance. So let's start with the first question here. The codon UAU codes for the amino acid tyrosine or TYR, and we want to use this as our wild type codons. Wild type means it's the normal one, the unmutated one, and we want to create the following mutations. Okay, a silent mutation, missense, as well as a nonsense mutation. So on the quiz, you will be given the codon table, which you will be able to use to create your mutations. Okay, so our wild type is UAU over here. So let's write that down, and you can see UAU codes for tyrosine as stated in the question. So if you want to create a silent mutation, you want to choose something that would code for the exact same thing. Okay, so you can see the codon UAC codes for tyrosine, just like UAU. So that will be the answer for the first question over here. Okay, silent mutation is UAC. Okay, you can see UAC, same amino acid, right? Um, I mean, the question didn't ask you to write the amino acid for the mutant, but I'm gonna put it there just to show you that it is the same. Now, a missense mutation is a mutation where the mutated codon codes for a completely different amino acid. So you want to choose something that doesn't code for tyrosine. The easiest way to create a missense mutation is to change the first nucleotide of your codon. You can change to anything you like. Okay, so instead of UAU, we can change it to AAU. Okay, so let's take a look at the codon chart and see what AAU codes for. AA, so I'm going to be looking at this box, and AAU codes for ASN, which is again like a different amino acid than the original, right? And that's why it's called a missense mutation. And finally, we want to create a nonsense mutation. A nonsense mutation is a mutation where the mutated codon is going to be a stop codon. Okay, so it's going to cause your translation process to stop prematurely, creating a shortened protein, or as it's called, a truncated protein. Okay, so there are three stop codons, which you should know by now. There is the UGA, UAA, and UAG, right? So we take a look at our wild type here, UAU, and we want to change it to one of these things by switching one of the nucleotides. So there are a couple of ways to do this. You can either change the last nucleotide to an A, or you can change it to a G. Right? Both will be correct answer. I'm just going to go with a UAA here, and that's going to create a stop codon. All right. That is how you mutate a wild type codon into each one of these. All right, next question here. We have a coding strand that's being given to us, and I already did the transcription for you, so you have the mRNA sequence here, and I did the translation as well. So we have the amino acid sequence. Okay, and now there is a mutated strand in the question, and it asks you to write the mutated protein sequence. Since this is DNA and it's coding strand, if I want to write a mutated protein sequence, I must first change it into RNA, right? And changing from coding strand to RNA, all you have to do is replace all the T's with U, right? And I'm gonna chop it up into triplet as well, so it's easier for me to keep track. Okay. And you can see once you did the translation that we are actually missing a nucleotide, right? Instead of like having triplets all the way, we only have two nucleotides at the end. So this suggests to us that something was deleted. Okay, it's probably a deletion mutation. Okay, so we're going to use the codon chart to help us translate the RNA sequence, starting with AUG. Of course, that is going to be MET, the starting codon codes for methionine. And we have CAA next. So CAA GA, which codes for GLN. We're going to go ahead and do AUC next. So AU and C, right? You should have your own codon chart as you are going through this review with me, so that you can try it out yourself, right? There's a GAU as well. So G is over here, A is over here, and U. So GAU codes for ASP, right? So that is it. Okay. I mean, the last two thing here is AA, which just doesn't code for anything. We were supposed to stop, but it doesn't stop. There is a new reading frame. There's no stop codon in a new reading frame. So it's just going to keep on going and going, until eventually maybe hit another stop codon. So describe the type of mutation that occurred, right? We noticed a nucleotide was missing, and that's how we know this is going to be a deletion mutation. Okay, and you can be a little bit more specific. You can compare the wild type with the mutated strand, and you can see, ATG, so no problem there. And then GAG, and here we have GAA, right? So the G is actually deleted, right? It's missing, right? So deletion mutation. I'm gonna say the second G in the coding strand is deleted. Okay, and what happens when you have a deletion mutation? Well, that's going to change the entire reading frame, right? And we call that a frame shift mutation, right? Caused by a deletion mutation. Okay, if you're deleting or adding nucleotides that are not multiples of 3, that will always result in a frame shift mutation. Okay? And by changing the reading frame, it leads to extensive missense. Okay, everything after the point of deletion is going to be wrong. Okay, and furthermore, the protein that we're making, it's much longer than what it's supposed to be because we missed the original stop codon by shifting the reading frame. Okay, so that's the answer for part B. There are more practice, right, in your study guide. I think there are a total of like 6 questions related to finding the mutations. We did some of this in class already. It's worth going through those again in preparation for the quiz. All right, so here is another question here. Create a point mutation in the coding strand. So you will create a truncated protein. Okay, so this is the same strand as question 2. It says it's the same strand here, which means, if you need to refer to the amino acid sequence, you could, okay, but you don't really have to. It depends what you're trying to do here. So this is the coding strand, and we need to create a truncated protein. Truncated protein means a shortened protein, right? And you will have a shortened protein if you create a stop codon before it's supposed to end. Okay, so we call this a nonsense mutation, right? Nonsense mutation. So there are different ways to creating the nonsense mutation depending on what the question is asking you to do. In this specific case, we have to do a point mutation. That means you change one of the nucleotides to create the nonsense. Okay, so this is a coding strand that's given to us. To make it easier, we should write down the stop codons, which again you should be quite familiar with at this point. It's UGA, UAA, and UAG, right? So this is again the RNA codon. And we want to think about what these codons are going to look like in the coding strand of a DNA, right? So the relationship between the RNA and the coding DNA is that you have to replace the uracil with thymine. So therefore, the stop codon in the coding strand should look like TGA, TAA, and TAG. All right. So all that we have to do is to look at the sequence that's given to us, and we see what letter can we change to create one of these stop codons. Okay. And if I mean we cannot do it at the end because that's like the original stop place. We have to do it like beforehand. So in this case here, right, CAG, pardon me, CAG, we can mutate the C and change it to a T, right? So that's gonna be a stop codon, right? When the cell does the transcription, the TAG will become the UAG, and then when they do the translation, right, you will start with methionine because that's AUG, and then it would stop. Right? So that would be an example of how you can create a truncated protein, right? Essentially, the whole protein is gone, right? You just have one amino acid and then you stop, right? So that's going to be a nonsense mutation caused by, again, a point mutation. Now, this one, we want to create an insertion mutation. So you have to add a nucleotide that would result in truncated protein. So same result. And the result is going to be a nonsense mutation, which causes your protein to become shortened. But how you create that is different. Right? In the previous question, we changed one of the nucleotide, but in this particular question, you have to be inserting a nucleotide, right? So you want to insert something so that you're going to have like TGA, TAA, or TAG. So there is a spot right here. If I insert a T, right, then the ribosome will read, AUG, CAG, and then UAA, right? So that's going to be the stop codon right there. Okay. Let's take a look at the next question here. It says, based on the pedigree below, what kind of inheritance pattern does the trait follow? Right? You must justify your answer to get full marks. All right. So remember, squares are like male, and then circles are female. And if they're shaded in, it means they are expressing the trait. Okay, they have the trait. And so here we want to state that genotype, the genetic makeup of individual number 1, as well as individual number 7. Right? So we must first determine what kind of inheritance pattern this trait follows. Is it a dominant trait or is it a recessive trait? Okay. So you see two individuals here. Let's just say we take a guess. Okay, we guess that this is going to be a recessive trait. Okay? If this is recessive trait, the only way for the person to express the gene is if they are homozygous, if they have little a, right? Yeah. So this person, individual number 1, has the trait. If we are assuming that this trait is recessive, then again, the genotype must be little a, and his spouse also will be little a. So if that's the case, if both parents are little a, and you do the Punnett square, 100% of their kids would also be little a, right? Because they can only pass on the recessive trait. But when we take a look at their kids, right, number 5 and number 6, they have the trait, which follows the logic here. But if you take a look at number 7, right, number 7 does not have the trait. So that tells you it's impossible for this trait to be recessive. Okay, for it to be recessive, you would expect number 7 to also express the trait, right? So, in fact, in class we talk about when both parents exhibit a trait and one of the child does not, that's a telltale sign that the inheritance pattern is dominant. Okay, so we will say this is a dominant trait. Okay, and the reason, both individual 1 and 2 exhibit the trait. We know they have the trait because they are shaded in, right, on the pedigree. But one of their children Right? Number 7 does not have the trait. Okay, so for that reason, it's going to be dominant. Right? Which means the person is going to be big A, small a. Okay, we know they're not big A because otherwise, again, the whole family would have it, right? The fact that they have one son that does not have the trait means they must be heterozygous. So individual number 1 must be big A, small a, and individual number 7 must be small a, small a. Okay, now just to refresh your memory, what would recessive look like? Okay, what would recessive look like? Recessive is going to be like for the condition to be recessive, right? Like a telltale sign for that would be you have 2 parents who don't have the trait, and then one of the kids have it. Okay, so that is what you're looking for. If you try to distinguish between dominant and recessive, that's what you're trying to look for. For dominant, parents both have it, but one of the kid does not. For recessive, it's the other way around. Both of the parents don't have the trait, but one of their kids do. Okay, at least one of their kids do. That's what I should say. All right. So in this case, big A, small a for the parents, and then small a, small a. All right, so that would be the telltale sign for recessive inheritance pattern. All right, moving on, we have number 6 here. This is a monohybrid cross question. We have a black guinea pig, which is dominant over the white coat color. So we're gonna put big B for black coat, and we're gonna put small b for white coat. Remember to use the same alphabet for the same trait. Okay, just use the capital letter for the dominant trait and the small letter for the recessive trait. And in this particular question, we have a hybrid black guinea pig, which is being crossed with another hybrid black guinea pig. Okay, which means the genotype of the parents are going to be big B, small b for both of them. Okay, so we're going to go big B, small b cross, big B, small b. We will set up our Punnett square. So what happens is when the guinea pig basically produce the gametes, right? These genes will these alleles will separate. So half of the gametes will carry the big B, the other gamete will carry the small b, and similar for the other parent. And so if this sperm fertilizes that egg, we're going to have big B, big B. Similarly here, we're going to have big B, small b, and small b, small b. The question asks you to calculate the expected percentage of both genotype and phenotype. So looking at this table, you can say for the genotype, there is going to be a 25% chance for big B, 1 out of 4 boxes, right? 50% chance of getting big B, small b, and 25% chance of getting small b, small b. So that's the genetic makeup. Now remember, the genotype is not always same as the phenotype percentage. Okay, so we have to write it out and see. Big B is black coat because capital B is dominant. Big B, small b is also black coat. So 75% will be black coat, and there's 25% chance that it's going to have a white coat. Okay, so that is a monohybrid cross question. All right, next we have a dihybrid cross question over here. Consider the color and position of the flower for a certain plant. It says capital R is representing red flower, and that's going to be dominant over white flower. Axial flower is dominant over terminal flower. Okay, axial means it is going to be, the flower is going to be coming out in the middle of the branch, whereas terminal flower means it's going to be coming out at the end of the branch. Okay, I mean, like I said, right, you don't necessarily have to know what it means to do the question. All you have to know is one trait is dominant over the other, right? Okay, so the genotypes of the parent generations are heterozygous red flower and pure axial, crossed with heterozygous red and heterozygous axial. And it says stay the phenotype. Okay, so because it's big R, it's going to be red. Capital A is going to be axial for this parent. For the other parent, because there is a capital R, so it's also going to be red, and it's also going to be axial. So phenotypically speaking, they look the same, but genetically their makeup are going to be different. Create and complete a Punnett square for the cross between the two parents. So to do this, you have to write out the gametes for each of the parents, right? So we start with a big R, small r, big A over here. And we will write the possible gametes, right? So we mix and match. We take the big R and we can combine it with the A here. So big R, big A is going to be one possible gamete. And if you do it again, big R, big A, it's the same thing. So we don't have to write it another time. Then you do it with a small R, big A. You're going to put it right here. And then you can do it one more time, and it's small R, big A again, right? You don't have to repeat the gametes. It's going to cut down your work substantially, okay, by not repeating yourself. So that's one of the parents. Let's take a look at the other one. The other one is big R, small r, big A, small a. And we're going to write down the possible gametes. So we have big R, big A. We have big R, small a. Small r, big A. And lastly, we have small r, small a. Okay, so this is all different. In this particular case, there are 4 possible combinations for the gametes. So if you take this parent and then you cross it with the other parent, we're gonna end up having a Punnett square that is 4 by 2. Right? So let's do that. Let's do a 4 by 2 Punnett square. Okay, so starting with we're gonna put the gametes of this parent over here: big R, big A, small r, big A. And I'm gonna take all these gametes from the other parent. We're gonna put it up here. So now we're gonna combine these gametes. Okay, let's say this pollen fertilizes that egg. We're gonna have maybe let me use a different color here. We're gonna get big R, big R, big A, big A. Next one is gonna be big R, big A, small a. And we just copy the letters, right? Big R, small r, big A, big A, big R, small r, big A, small a, and so on and so forth. You just complete the Punnett square like we've done in class. All right, that's it. Right? The question says create and complete a Punnett square, right? Then the next question asks you to state all the expected phenotypes and their expected percentages for all F1 offspring. So this is F1, the first generation, right? And it's only asking for phenotype. So what we can do is just look at the first one. First one is big R, big A, big A. So that's going to be red and axial. And you want to find everything else that's also red and axial. Okay, so everything that has a big R and a big A. So that's red and axial here for these one as well. Big R, big A here, over here, right? That's all, right? So all these 1, 2, 3, 4, 5, 6, 6 out of 8. 6 out of 8. It's a good idea to reduce it. Which is what 6 out of 8. 6 out of 8 is going to be 3 over 4, right? 3 over 4. You divide the top and the bottom by 2. And you can bring your calculator to the test, to the quiz if you want. 3 divided by 4 is going to be 75%, right? So 75% of the F1 is expected to have red axial flower, right? And the other ones, little r, that's going to be white and axial, right? And this one is also white and axial. Let me just double check if it's yeah, little r is white. Okay, so that's 2 out of 8 boxes, which if you reduce is 1/4, so 25% is going to be having white and axial flower. Okay, so that's a dihybrid cross questions. Again, tons of practice available in the study guide. Make sure you look over those. Okay, last question here. In a certain flower species, missing an S here, species, the allele for red flower is incompletely dominant over the allele for white. If it's incomplete dominant, it means the heterozygous condition is going to be some kind of blend in between, right? Compared to the pure genotype. So in this case, we have the red flowers, which is going to be big R, big R. We're going to have little r for white, and the heterozygous genotype is going to look like pink, right? The question tells you so. 2 pink flowers are crossed and produced 100 flowers. How many of those flowers are expected to be pink? So 2 pink flowers means we are taking big R, small r, and we crossing it with another big R, small r. And when we fill in the Punnett square, we can see that there are 50% chance that we're going to produce an offspring with big R, small r, which means there's 50% chance it's going to turn out to be pink, right? So if they produce 100 flower, 50% of 100 is 50, right? So you would expect to have 50 pink flowers. And that's how you do it. Okay. So that's it for the review. I hope you found this useful. And again, make sure you go through these questions and try to do them on your own. And if you have any questions, please feel free to email me. All right, that's it for now. Thank you for watching, and good luck on your quiz.